Hello everyone, welcome to the world of microbiology. In this video, we are going to discuss the human virus named chickenpox and plant virus named cassava bean mosaic virus. But of course, before that, let me first read to you the objectives. At the end of the lesson, you are expected to define the varicella zoster and cassava bean mosaic viruses, explain the pathogenesis of varicella zoster and cassava bean mosaic viruses, and demonstrate through role play the impact of varicella zoster and cassava bean mosaic viruses to all living organisms. Let us start from the human virus named varicella zoster virus or chickenpox. Chickenpox, also known as varicella infection, is the primary infection caused by varicella zoster virus. It is a highly contagious disease, most commonly seen in children under 10 years old. Chickenpox is usually a mild, self-limiting illness, and most healthy children recover with no complications. Adults tend to suffer more severe diseases than children. Characteristics and classification. Varicella zoster or VZV is an alpha herpesvirus and a member of Diplodocnaviridae realm, Hongongviridae kingdom, Piploviricotta phylum, Hervivicitis class, Herpes viralis order, Herpes viridae family, and Varicellovirus genus. As you can see in the structure, this virus is a double-stranded DNA virus, and each genome contains at least 70 genes and 41 core genes. VZV also encodes at least 3 immediate early proteins that are located in the tegument of variants and regulate virus transcription. Varicella zoster virus causes chickenpox infections. Most cases occur through contact with an infected person. The virus is contagious to those around you for 1 to 2 days before your blisters appear. VZV remains contagious until all blisters have crusted over. The viruses can spread through saliva, coughing, sneezing, and contact with fluid from the blisters. Furthermore, varicella is characterized by mild headache, fever, malaise, eruption of blisters on the skin, and eruption mucous membranes. The long incubation period, usually around 15 days up to 21 days. Infected period between 1 to 2 days pre-eruption and 5 days post-eruption. Pathogenesis. The portal of entry of virus is via the inhalation of respiratory droplets through respiratory tract or also via the contact with infected persons, as what I have discussed earlier. The virus infects the upper respiratory tract. The virus replicates and proliferates in regional lymph nodes of the URT. This follows the primary viremia in the bloodstream. From the bloodstream, the virus localizes in the skin, mucous membrane, and other organs, particularly the liver and spleen, which results in the secondary viremic stage. This stage is manifested by the appearance of characteristic rushes composed of macules, which rapidly develop into fluid-filled vesicles. Within 3 days, the fluid becomes cloudy with the influx of leukocytes, fibrin, and interferon. The pustules then dry up, scrubs form, and they desquamate. Lesions and vesicles are present in all stages then after. The virus-infected host cells are cleared by T-cell mediated immune mechanism and antibody-dependent cell-mediated cytotoxicity or ADCC. The virus may also enter nerve ending and transport to dorsal root ganglia, where it remains dormant in sensory nerve ganglia, dorsal root. After infection, the virus remains latent in cranial nerve and dorsal root ganglia, with reactivation causing dermatomal or in the immunocompromised host disseminated zoster. Prevention and control. Preventing Varicella in healthcare settings is done by following standard precautions, plus airborne precautions, negative airflow rooms, and contact precautions until lesions are dry and crusted. The best preventive measure for chickenpox is the chickenpox vaccine. The vaccine is a live attenuated vaccine administered subcutaneously. 2 doses of the vaccine are about 90% effective at preventing chickenpox. Ecological or medical impact. Varicella zoster virus serves as an economic burden to any country because of its cost in terms of hospitalization and even in the cost of its vaccination. In contrary, these serve as a medical advantage in medical field primarily because of the sales of vaccine and paid service in hospitalization. And now let us move on and proceed to the plant virus named Cassava mosaic virus. By the way, what do you think about mosaic virus? Okay, mosaic virus is the umbrella name for a variety of viruses that attack your plants. What's dangerous about it is that mosaic virus attacks your plant at a cellular level by tracking down the chloroplast, which is the site where your plant cell absorbs sunlight and transfers this into energy which your plants need to live. On this note, cassava mosaic virus, commonly known as CMV, is one of the viruses that leads worst disease to cassava. It affects the productivity of the plant and a widely occurring virus to all cassava that seemingly cause cassava mosaic disease. Characteristics and classification. Cassava mosaic virus is a circular single-stranded DNA virus and a member of Monodnavirium, Pseudocouveri kingdom, Corisna vericota Ripensivirices class, Giplafoviralis order, Geminiviridae family, and Bugumu virus genus. Pathogenesis. Cassava mosaic disease or CMD could be spread by the self-feeding white fly called Bemisia tabaci. This fly acts as a vector or an agent of carrier or of pathogen. CMV, moreover, could also spread through vegetative propagation or use of cutting from infected plant, mechanical transmission through sap, seeds, and pollen, and lastly, biological transmission by nematodes and other insects. After the plant is infected, it will then likely experience mosaic symptoms such as mottling, discoloration, malformation, and puckering of the leaves, clearing, bundling, and thickening of the bean, and entire plant becomes stunted. Prevention and control. Controlling insect vector by spraying insecticide. When using an insecticide, always wear protective clothing and follow the instructions on the product label, such as dosage, timing of application, and pre-harvest interval. Removing and burning affected plants. The uprooted plants should be carried away from the field and exposed to the sunlight for drying and then burned to kill the viruses. Do not bury the diseased materials in the field, for it will not kill the virus or viruses. Proper use of cultural instruments. Prepare healthy cassava cuttings which will be used to replace the diseased cassava plants. Changing planting season. Cassava planted after the beginning of rainy season is less susceptible to disease. Use of resistant varieties of suppressing pathogens activity. Ecological or medical impact of the virus. This virus impact agricultural damage, and it also loss or reduce the production, which is another term of farmers' deficiency. All in all, this virus is a pathogen. It serves as a burden in production, and it also serves as a burden to all farmers. That's all for today. Thank you and God bless.